Good afternoon, ladies and gentlemen, and welcome to the first of this series of webinars on glyphosate. These seminars are being funded by the Glyphosate Task Force, and it's my pleasure to be your hoster today. And my name is Caroline Harris. I am the Corporate Vice President and Principal Scientist at Exponent International in the UK. My background, I'm a specialist in pesticide residues and consumer risk assessment. And while I don't do any work on toxicology or epidemiology, I make extreme use of the information that is provided by my colleagues. The first of this sessions will look at the carcinogenicity and epidemiological research on glyphosate, and we'll focus on the evidence that is available to us. We have two outstanding speakers for you today, and we're starting off our session with Dr. Wallace Hayes, who will be known to many of you as a toxicologist that's been working with industry and government and academia for over 35 years. Wally is currently holding appointments at Harvard and also the University of Massachusetts. And this afternoon, Dr. Hayes will be focusing primarily on the evidence available in the database, which is extremely large, relating to the long-term toxicology studies with glyphosate. Dr. Hayes, I'll pass over to you. Thank you.
Thank you, Carolyn. Let's see if I've got control. I hope I do now. For those of you joining the webinar, welcome. It's in the morning in the U.S. For those of you in Europe, it's late afternoon, and we're just glad that you're with us. We're going to spend the next 30 or so minutes just looking at the information that is available related to the long-term studies, carcinogenicity studies dealing with this compound of interest to all of us. And what we're going to be talking about is just take a minute to remind ourselves of what is glyphosate. Then we're going to look at the basis that the International Agency for Research on Cancer based its classification on, and part of that will have to do with genotoxicity and oxidative stress. And for those of you that don't know, there will be another webinar on November the 10th, which will go into detail on both of these areas. And then we'll spend the rest of our time talking about the studies that have been reported in mice and rats and the various types of tumors that have or have not occurred within these studies that have been done in these animal species. So if we ask ourselves the question, what is the chemical that we're looking at? It's really a very, very simple chemical, partially glycine, which is a normal part of proteins, and then the moiety that makes up the other part of this simple molecule. And if we think about it from the standpoint of its activity as a herbicide, it's non-selective, it's a broad-spectrum herbicide, and it works by preventing plants from making aromatic amino acids needed for plant growth. And it's really that area of selective toxicity that entomologists and herbicidologists have been doing for a long time, looking for mechanisms that really don't exist in higher mammalian forms. So when we look at the Iarc monograph, which came out in 2015, volume 112, the suggestion was that there was strong evidence supporting that glyphosate can operate through two key characteristics of known human carcinogens, and that's genotoxicity, 1, and 2, induction of oxidative stress. And again, I would just remind you about the webinar that will be taking place on November the 10th, which will deal in much greater depth on these two characteristics. When we think about their outcome of this particular review that they did, they used genotoxicity to support their classification as a probable carcinogens to humans, putting it into the group 2A classification. As we look at the data that surrounds genotoxicity, and I'll say just a little bit about metabolism, when you look at the molecule, and Iarc certainly states this, that it's not an electrophilic molecule, so you wouldn't expect it to interact with neutrophilic type substances. And if you do a very simple structural alert analysis using software such as Derek, you don't get any structural alerts which are suggestive that genotoxicity, particularly mutagenicity, is probably not there, or if there are genotoxic effects, they may be secondary to toxicity or resulting from mechanisms other than DNA reactivity. And just very simply, you can look at the molecule, it's polar, it's not very readily absorbed, there's limited biotransformation, and it rapidly is excreted unmetabolized. So when you just think about this for a moment, you've got a molecule that's not particularly reactive, it's not particularly well absorbed orally, it doesn't get biotransformed to any great extent, and it's in and out of the system rapidly. So when you think about it from that standpoint, you have to ask yourself the question, if it is genotoxic, how does it do this? And you have to tune in on November the 10th to really get the full insight into that. But one can look at the overall evidence, and particularly the large number of Ames assays that have been done, which support the fact that the compound just does not induce gene mutations. And if you take the JMPR 2016 conclusion, which is on your screen, and I'll just read it to you, it says the overall weight of evidence indicates that administration of glyphosate and its formulation products at doses as high as 2 grams per kilogram body weight by the oral route, which is the route most relevant to human dietary exposure, was not associated with genotoxic effects in an overwhelming majority of studies conducted in mammals. Again, the statement, a model considered to be appropriate for assessing genotoxic risk to humans. So when you take that overall conclusion and use the JMPR, the outcome says it's probably not a genotoxic material, and it certainly does not appear to induce gene mutations. There's really no evidence of integrated data analysis using mode of action, and when you think about human mode of actions for carcinogens, we're really talking about 4 things: DNA reactivity, increase in cell proliferation, immunosuppression, and that only has to do with viruses, and estrogenic activity. And oxidative stress falls in some place in there, but if you look at the data related to oxidative stress, it seems that unless you look carefully at the literature, you tend to bend it. The relationship to other key characteristics, when people, particularly Iarc, was looking at the oxidative stress, a lot of the other key characteristics were not considered, but I think the important thing was the idea of dose, temporality, cohesion, consistency, and target organ relevance just were not addressed in the Iarc document. Again, I would remind you that the details of both of these will be on the webcast on November the 10th. So let us now get into what is going on within the European Union as far as the renewal evaluation for glyphosate. The company submitted their data back in 2012. The renewal assessment report was presented by the Germans. This went into the European Food Safety Agency in cooperation with all member states, and it was peer-reviewed. And it was about the same time that the Iarc monograph was published in March of 2015. The European Food Safety Agency's final conclusion report came out in October of 2030, and we're sitting now waiting for the European Commission to decide whether to include the substance in their list of approved active substances. Now, as we begin to look at the types of reports that have come out, particularly from the European Union, we know that Germany, the European Food Safety Agency, and member states have concluded that the chemical is unlikely to pose a carcinogenicity hazard to humans, and the overall evidence just does not support classification with regard to its potential carcinogenic activity. In addition, the German Federal Institute for Occupational Safety and Health has posed no classification for carcinogenicity to the European Chemical Agency in Finland. If you then begin to move outside of the European Union and think about and look at the regulatory reviews that have taken place recently, they've all concluded that glyphosate is unlikely to pose a cancer hazard to humans. Australia and New Zealand reports came out in 2016. There was a joint FAO-WHO meeting on pesticide residues in 2016. The Food Safety Commission of Japan's report came out in 2016. The European Union, as well as EFSA, came out with their reports, and in Canada in 2015, their report also came out. And if you look to the right of the slide, you can see that in all of these reports, glyphosate does not pose a cancer risk to humans, is unlikely to be carcinogenic to humans or genotoxic, unlikely to pose a carcinogenic risk to humans, does not support classification with regards to its carcinogenic potential, according to regulation number 1272/208. So as you look around the world from regulatory bodies, it's very, very apparent that glyphosate is given a clean bill of health regarding its carcinogenic potential to humans. In addition to these regulatory bodies, there have been a number of peer review publications that have supported the fact that it is unlikely to pose a cancer hazard to humans. And three of these, one came out in 2015, the other two appeared in the peer-reviewed literature in 2016. In the 2015 Grimes paper, they looked at 14 chronic carcinogenicity rodent studies and concluded that the incidence just simply is not there to support it being a carcinogenic chemical. That was in critical reviews of toxicology. The Turon publication was in the European Journal of Cancer Prevention in August of this year, and again came out with a similar type of review of the Iarc document. And then the Williams document, which was peer-reviewed and appeared in critical reviews of toxicology this year, along with several other publications in that supplement on animal bioassay, genotoxicity, epidemiology, and human exposure. The overall conclusion, not only of the Williams paper, but all three of these papers, is that glyphosate is unlikely to pose a cancer hazard to humans. So we have regulatory bodies supporting the results of this peer-reviewed literature that's just recently come out. The industry, I guess, was not satisfied at that point, and they decided to bring together an expert panel, and they got Intertech Scientific and Regulatory Consulting Services out of Canada to bring together an expert panel, and this was composed of 16 really first-rate scientists from the U.S., Canada, Brazil, the United Kingdom, and Germany. And they had 4 subgroups: exposure, epidemiology, animal bioassay, and mode of action, genotoxicity. I was not part of that group, but subsequently been able to review the data that we'll be talking about shortly. This group looked at information that Iarc did not review, including regulatory GLP studies. And according to the Williams et al. Paper, their conclusion was really fourfold: that there was a lack of statistical significance, strength, and consistency. There was a lack of a dose-response pattern. The tumors did not progress to carcinomas, and the high-dose instances, for the most part, all fell within the historical control range of the various laboratories where the test took place. Well, Iarc, as all of you know, did indeed classify glyphosate as a probable carcinogen to humans, a group 2A. And this was based, in their minds, on the weight of evidence that glyphosate is considered to indeed pose a risk, and they placed it in the 2A category. This classification by Iarc is inconsistent with all of the information that I've given you by other WHO expert committees, by regulatory agencies around the world, and by these third-party experts who have looked at not just the information that Iarc looked at, but the regulatory submissions as well. And their conclusion in all of these cases, regulatory, WHO expert committees, third-party experts have concluded that the weight of evidence says that glyphosate is unlikely to pose a cancer hazard to humans. Now, the basis of the Iarc classification, I've just shown you a slide here that indicates there's some epidemiology information that they think is limited, and you're going to hear after this a really first-rate discussion on the epidemiology. They suggested that the animal evidence was sufficient, and they proposed a mode of a mechanism of action, a mode of action, dealing with genotoxicity and oxidative stress. And again, remember, on November the 10th, you can hear more about both of these series of endpoints. And they concluded that glyphosate was a group 2A carcinogen. Now, Iarc's criteria for classification really falls into 3 buckets. The first is you need to have sufficient evidence in one of these 3 buckets, preferably in all 3 of the buckets. The first one suggests that you can have an increased incidence of malignant neoplasms or of combining benign and malignant neoplasms in 2 or more species or 2 or more independent studies in one species carried out at different times or different laboratories under different protocols. Or you can have an increased incidence of tumors in both sexes of a single species in a well-conducted study, ideally conducted under GLP practices, or thirdly, a single study in one species and sex where you have malignant neoplasms occurring to an unusual degree with regard to incidence, site, type of tumor, or age at onset. You're going to see a couple of things as we get into the individual studies. First of all, the third criteria simply was not met in any of the studies. There was no unusual degree of neoplasms. The second one, I would suggest to you, in both sexes of single species also did not occur. So really the evidence needs to fall in that first category. And if you'll also notice, the word trend does not occur in any one of these 3 criteria that's there. They talk about an increased incidence, or a unusual degree. Nothing about trend. Keep that in mind as we move forward. Now, Iarc concluded that glyphosate induced a positive trend in the case of renal tubule carcinomas, a positive trend in the case of hemangiosarcomas, an increased incidence of pancreatic islet cell adenomas, and an increase in hepatocellular adenomas in male rats, and a trend in the case of adenomas in females. And you notice that 3 of these are trends, with only 2 being a so-called increased incidence. And if you think back to the criteria, remember the word trend was not there. If we look at and line up regulatory bodies from 1987 to 2016, and we look at the 4 primary studies, 3 from Monsanto and 1 from Chemnova, you'll see the only place where there is red is with the WHO Iarc report. They're the only ones that suggested that there might be a carcinogenic effect. All the other regulatory bodies that looked at this have concluded in a different fashion from Iarc's approach to the issue. Now, if you look at the way studies were evaluated, there's some very basic differences. I've listed in the left-hand column the types of differences regarding the evaluation, and then Iarc and the subsequent expert and regulatory reviews that took place. If you look in the right-hand or the left-hand column, it says review primary study reports. Iarc did not review the primary study reports, whereas the other folks did. Dose response, not really considered at Iarc, the others did. Certainly Iarc heavily depended on trend analysis. The other folks did look at that, but concluded that they were negative. Review the pathology reports or the pathology working group reports. Iarc did not. The other folks did. Considered histopathology of pre-neoplastic findings for treatment-related induced carcinogenesis. Iarc did not. The other folks did, and there was no evidence of pre-neoplastic progression. Evaluate same neoplastic studies in the same similar design in same species. Not really, and you're going to see where they pulled out one set of cancer types and did not look at another set, whereas the regulatory reviews did. consistent with previous expert regulatory reviews. Certainly, Iarc stands out alone, and review consistent with OECD Guidance Document 116. The answer is no. And again, in the case of the experts and regulatory reviews, they evaluated statistics, biological plausibility, dose response, and the pathology as you would expect it to be done. Now, let's look at the tumor types that were suggested by Iarc to be present. And we'll look first at the renal tubular tumors in male mice. This is one of the Monsanto studies. It was conducted, or was concluded tumors were spontaneous and not related to treatment. If you look at the numbers, they're adenomas 0, 0, 1, and 3. And the study was then re-looked at by a well-known renal pathologist, Dr. Kirshner, and he verified the tumors in the high and mid dose, but he identified a small renal tubular neoplasm in one control animal. And if you look then at the combined instances, it's 1, 0, 1, and 3, and there's no statistical difference pairwise or trend. And this was not included in the Iarc evaluation. If you look at the history of the U.S. Environmental Protection Agency review of the renal tubular neoplasm, you go from the pathology working group to the FIFRA scientific advisory panel to the health effects carcinogenicity review committee, and you see that the overall conclusion going through this pathology very, very carefully, the conclusion by EPA was that it should be classified as a group E, which signifies evidence of non-carcinogenicity in humans. In some cases, I think, and here's an example where there was selective data reporting. Remember, the renal tubular neoplasm in the second mouse study was not reported by Iarc. And so if you combined all of these, that is the two studies, you've got 2, 2, 0, and 0, which says there's no dose response. As I've said, Iarc 112 does not mention these conflicting data. These data were supplied to Iarc, but they just were not considered as part of the overall evidence. And when you see it, it's certainly there. Hemangiosarcomas in male mice. If you look at the data here, it's 0, 0, 0, and 4 out of 50 or 8%. These are CD1 mice were fed these diets at dose levels of 100, 300, and 1,000 milligrams per kilogram. And when the study was reviewed initially, it was concluded no statistical significant increase in any tumors. Again, the first mouse study was not reported, and if you look at it, there's no dose response, and in fact, there are no malignant tumors present in these studies. The data was supplied to Iarc, it was not mentioned, and there was just no progression to a malignant 0, 0, 1, and 0. If we go to pancreatic islet cell tumors in male rats, we find that the Monsanto study concluded early on there was no increase in tumors considered treatment-related, 0, 5, 2, and 2. Again, the doses were 310 and 31 milligrams per kilogram per day. These are adenomas, and here again, we don't find any migration into carcinomas. The second study, there was one in the control 8 in the low dose, 5 in the mid-high dose, and then 7 in the high dose. And the dose levels were 0, 89, 362, and 940. Now, if you look at the EPA conclusion of these studies, they stated that there was no statistical significant positive dose-related trend in the occurrence of these tumors in males, no progression to carcinomas, and the incidence of hyperplasia was not dose-related. Therefore, the pancreatic islet cell tumors were not considered to be compound-related. The incidence of pancreatic islet cell tumors for the two studies does not show a dose-response increase in adenomas or adenomas carcinomas combined is within the range of open literature controls for male Sprague-Dawley rats, which runs from 0 to 17%. So the EPA, U.S. EPA concluded that the chemical should be classified as a Group E, evidence of non-carcinogenicity for humans based on lack of convincing carcinogenicity evidence in adequate studies in two animal species. The hepatocellular adenomas in male mice, again, looking at the data, the doses were 89, 362, and 490. In the Stout and Riker study, the authors concluded that an oncogenic effect was not observed in this study. And if you look at the adenomas, 3, 2, 3, 8, no progression to carcinomas, 3, 2, 1, and 2. So again, the EPA's conclusion was despite the slight dose-related increase in hepatocellular adenomas in males, this increase was not significant in the pairwise comparison with controls and was within the historical control range. They go on further to say, furthermore, there was no progression from adenomas to carcinomas, and instances of hyperplasia were not compound-related. Therefore, EPA concludes the slight increased occurrence of hepatocellular adenomas in males is not considered compound-related, and as such, should be classified as a Group E, evidence of non-carcinogenicity for humans. The thyroid C cell adenomas in female mice, the data were 0, 113, 457, and 1183 milligrams per kilogram per body weight. This was a Stout and Riker study, and the authors concluded no statistically significant difference reported in incidence of thyroid C cell neoplasia. Again, if you look at the adenomas and compare them to the carcinomas, there was no migration from adenomas to carcinomas. 2, 2, 6, and 6, 0, 0, 1, and 0. Again, looking at the U.S. EPA's conclusion, and I quote, although C-cell adenomas slightly exceeded the historical control range for both sexes, there was no statistically significant trend or pairwise comparison with controls in males. In females, the incidence was not statistically significant in pairwise comparison with controls, but did have a statistically significant positive dose-related trend. However, EPA goes on to say there was no progression to carcinomas in a dose-related manner, and no statistically and no significant dose-related increase in severity of greater incidence of hyperplasia in either sex. Therefore, these adenomas in males and females were not considered compound-related, and EPA, the U.S. EPA, concluded, again, it should be a Group E, evidence of non-carcinogenicity for humans. So if you look at the overall carcinogenicity data for glyphosate, and you look at the weight of evidence across the board for all of the mouse and rat studies evaluated, the conclusion should be that glyphosate is not a carcinogen in rodents when dosed at levels up to more than 4 grams per kilogram body weight per day. And that's based on lack of statistical significance when appropriate statistics were applied. And the trend analysis that Iarc did was not significant according to the U.S. EPA cancer guidelines or the U.S. FDA guidelines for trend analysis. There was a lack of a clear dose-response relationship. There was lack of consistency throughout all mouse and rat studies. For the most part, the instances were within the historical control range of the laboratory. There was no progression of adenomas to malignancies. And those increases that were seen, for the most part, were beyond the limit dose of 1,000 milligrams per kilogram per day. The lack of relationship with pre-neoplastic lesions was also there. I told you about the lack of genotoxicity and the lack of no plausibility for a mechanism of action, including oxidative stress. So as the overall conclusion, I think the simplest thing is that Iarc's opinions conflict with the vast majority of the data that are out there, with the conclusions that have been reached by regulatory authorities, scientific bodies, and third-party experts who have for decades concluded that glyphosate is not a carcinogenic hazard for humans. The extensive animal carcinogenic data set is a strong basis for, I would suggest to you, concluding a lack of carcinogenic potential for glyphosate. So if you look at all of the data, and including the epidemiology, which you're going to hear shortly, the weight of evidence, I would suggest to you, clearly supports that there is no scientific justification to classify glyphosate as a human carcinogen, or for that matter, a carcinogen in experimental animals. Thank you very much, and I think we'll now have our epidemiology presentation, and then at the end, we should have some time for questions.
Thank you very much, Wally. I'll just remind everybody that you, although you're not able to join the audio for this webinar, you are able to pose us questions via the question link that was pointed out by the webinar organizer. We've got a couple of questions that have come through now, Wally. Would you like to take them now?
It's fine with me if, unless you want to go ahead with John and we do it at the end. I have no problems. Either way.
Okay.
It's your call, Carolyn.
Okay, so I'll just give a reminder to everybody that there is a facility to ask questions. We don't have a great number at the moment, so we'll hold on to the end of the session. But please feel free to ask questions as we go through. So we'll move on to our next speaker of the afternoon, who is Dr. John Acquavella, who is currently the Professor of Epidemiology in the Department of Clinical Epidemiology at Aarhus University in Denmark. Dr. Acquavella has held a number of posts over the years in academia, in government with the US EPA, and in industry as well. So this afternoon, John will review the data on the epidemiological research relating to glyphosate and give us an overview of the findings. Thank you very much, John.
Thank you. And good afternoon to our European audience, and good morning to those of you listening in the United States. I'm glad to follow Dr. Hayes' presentation and just relate to you the evidence from applicated biomonitoring studies, and then also to go through the epidemiology. I was the chair of the Intertech Epidemiology Committee, so I'll be drawing on that review, as well as the Iarc review and the review by the EPA that's just recently been made available. And I'll say, compared with the toxicology, the points of agreement are many in the epidemiology. The points of disagreement are very slight, and so I'll just be reviewing those. And then finally, I'll give you a little weight of evidence summary of the glyphosate epidemiology. Dr. Hayes covered most of this, but there's pretty general agreement that glyphosate has the exposure properties of having low vapor pressure and low dermal penetrability. Once absorbed, glyphosate is excreted rapidly without much metabolism. There's no potential for bioaccumulation. And measuring the parent glyphosate molecule in Europe provides a reliable measure of how much glyphosate has actually gotten into the body. And I've given you a couple of references at the bottom of the slide that go into these points in much more detail than I will today. And just as an example of the biomonitoring data, and considering a study that actually biomonitored much more extensively than any of the other human studies that have been done to date, I'm going to talk about the Farm Family Exposure Study, which was a study that was sponsored by a number of pesticide companies under the auspices of the Epidemiology Committee of the American Crop Protection Association. And the goal of this study was threefold, really. First was to try to understand how much exposure an applicator would get from a single pesticide application. And in addition, to find out how much exposure the spouse and the children on the farm would get from these applications. And the study looked at glyphosate, 2,4-D, and chlorpyrifos. The urine collection was very extensive compared with a lot of other biomonitoring studies. There were 5 days where 24-hour urines were collected. We used the terminology day -1 to be the day before the application. Day 0 was the day of application. And there were 3 days afterwards, which we were collecting urines. And if you read the paper by Baker et al. That I cite at the bottom of this slide, you'll see for the applicators there was very high compliance with the 24-hour urine collection. The glyphosate applications in this study were substantial. A third of them were 10 to 45 acres. A third of them were 46 to 124 acres, and a third were 125 acres to 439 acres. Farmers filled out a questionnaire at the end of the day about their application practices, whether they used rubber gloves, how many times they mixed and loaded, whether they had breakdowns and repairs that they had to do during the application, et cetera, kinds of factors that we think would help determine the likelihood of their exposure. And then we also had trained field observers who recorded the actual application practices that the farmer did. And I refer you to the publication by Acquavella, 2004, that goes into a lot more detail about the glyphosate results from this study. So this is a summary of the geometric mean glyphosate urinary values on the day before the application, the day of the application, and for 3 days after the application. We had an analytic method with a limit of detection of 1 part per billion. And interestingly, 40% of the applicators did not have a value that was greater than the limit of detection, while 60% did. A fair number of the applicators who had urinary values below the limit of detection had applied glyphosate to 100 or more acres. Geometric mean value peaked on the day of application at 3 parts per billion, and then it rapidly decreased back to the limit of detection. We did have one outlier value of 223 parts per billion. There was a right skew to the information, the urinary levels. Next highest value was in the 70 part per billion range. So there's one application where it seemed a lot of things went wrong, and that person had 223 parts per billion for their day 0 urine concentration. So when you just look at a a graph scaled to show you the glyphosate values. You don't get a sense necessarily of the exposure properties of glyphosate compared with other pesticides. But on this slide, I've just graphed the results for glyphosate along with the results for 2,4-D, which is the blue line, and metabolite of chlorpyrifos, which is the maroon line. And you can see while glyphosate peaked on the day of application and was excreted rapidly, both the metabolite of chlorpyrifos and the 2,4-D parent peaked on the day after the application, and then the excretion was more gradual than it was for glyphosate. One other perspective that I can give you is just to look at the milligram kilogram per kilogram values for all of the applicators for the 3 different chemicals. And you see on the x-axis here we have milligrams per kilogram, and on the y-axis we have the cumulative percent. So at the intersection of the x and y-axis here, we have the 0 percentile value for glyphosate. We trace that all up to the 100th percentile value for glyphosate. Likewise for chlorpyrifos, likewise for 2,4-D. So you can see that the glyphosate distribution is very compressed, and in fact it doesn't overlap that much with the distributions for the other chemicals. And 90% of the values for glyphosate were 0.001 milligram per kilogram body weight or less. One other perspective on the glyphosate biomonitoring is to see where the values for applicators are compared with regulatory guidelines and the toxicology studies that Dr. Hayes was talking about. And in this little box and whisker figure here, the colored area is the 10th to the 90th percentile values. So you can see the 90th percentile value is about 0.001 milligrams per kilogram. And you can see that's several orders of magnitude below the reference dose for EPA and other regulatory doses, and a couple of other orders of magnitude below the toxicology doses. Also need to bear in mind that herbicide application only happens a few times a year, so this exposure that we're diagramming here doesn't occur daily, doesn't occur weekly, doesn't occur monthly. Survey data tells us that on average applicators apply 3 or 4 pesticides a year for a total of somewhere between 10 and 20 days. So this is an exposure that happens a few times a year, and these are chronic exposures represented by those standards and the dosing and toxicology studies. So the reason I talk about what we know about applicator exposure before I go into the epidemiology is I think that by understanding the range of milligram per kilogram values for applicators, that will eventually help us tie together the epidemiology with what we know from other disciplines. So the biomonitoring data, to my way of thinking, establishes some basic considerations about biological plausibility in human populations. Mentioned glyphosate is used infrequently, and when glyphosate is applied, the applicator dose is much, much less than regulatory limits and much, much less than chronic milligram per kilogram doses in toxicology studies. So let's move on, and I'm just going to talk about 3 recent reviews. These are the most extensive reviews that have been done of the epidemiology in the past couple of years. There's the Iarc review, there's the Intertech expert panel epidemiology review sponsored by Monsanto, and then there's the recent EPA review. All 3 reviews agree that the data suggest no association between glyphosate and solid tumors, leukemia, or Hodgkin's disease. And all 3 reviews agree that the data for multiple myeloma are inadequate for evaluation. Multiple myeloma and non-Hodgkin's lymphoma were the detailed focus of the Iarc evaluation, the detailed focus of the Monsanto expert panel evaluation, and were a focus of the EPA evaluation. So the one point of moderate disagreement has to do with the evaluation of the non-Hodgkin's lymphoma results. So Iarc's conclusion was that there was limited evidence and that a positive association had been observed for non-Hodgkin's lymphoma. And limited in Iarc terminology means that a positive association has been observed in one or more studies, but chance, bias, or confounding could not be ruled out with reasonable confidence. The Monsanto expert panel concluded that the review of the glyphosate epidemiology literature and the application of commonly applied causal criteria does not indicate a relationship with glyphosate exposure in non-Hodgkin's lymphoma. And EPA was kind of halfway between Iarc and Monsanto in what I thought was a very excellent review. They considered that there were two studies that were high enough quality to be informative. One showed a positive association, one didn't show a positive association, and so they considered them to kind of cancel each other out. And they said a conclusion regarding the association cannot be determined based on the available data. So I'll just talk through the way the individual evaluations were done in light of the studies that were considered and try to show you where each of the groups came down in terms of their final evaluation of the evidence. So if we take the most recent report on study populations, some of them have been studied by more than one author. So here I've just taken the most recent reports. There are actually 7 epidemiology studies for non-Hodgkin's lymphoma and glyphosate that have been considered by the review bodies. I think the thing you noticed right off the top is that all but one of them were case control studies, and one was a cohort study. And here I've just detailed the locations where the individual studies took place and the years for which the subjects of the study provided their information and their risk for non-Hodgkin's lymphoma was evaluated. So there are 2 basic paradigms then for studies that have been done for glyphosate. One is a cohort study and one is a case control study, and I'll just briefly review how those studies are conducted for those of you who aren't epidemiologists, and this will be a very high-level review. So a cohort study looks the most like an experiment of any epidemiology study, with the exception that there isn't random assignment of the exposure by the investigator. So the way those studies go is that you identify study participants, and in this case, since there's only one study, we're talking about the agricultural health study. They've identified about 55,000 licensed applicators in Iowa and in North Carolina. The participants provided their exposure histories at enrollment into the study, and then the investigators followed them prospectively to identify health outcomes. In this case, I'll just be talking about non-Hodgkin's lymphoma. And then it's a pretty straightforward calculation. You calculate what we call the rate ratio, and we'll look at the rate of non-Hodgkin's lymphoma in licensed applicators who have a history of exposure to glyphosate, and we'll divide that by the rate of non-Hodgkin's lymphoma among licensed applicators who don't have a history of glyphosate use. And if this ratio turns out to be 1.0, we say that there's no association between the exposure and the outcome. Now, pesticide epidemiology studies work kind of backwards, but are nonetheless valid when conducted without major sources of systematic error. The way those studies work is that you identify cases who have the disease of interest. So in this case, non-Hodgkin's lymphoma, and usually it's from a cancer registry. So all of the case control studies for glyphosate used a registry of one sort or another, and the vast majority of them were able to histologically confirm all of the cases that they considered in their evaluation. You then move on to select population controls that are supposed to represent the source population for the cases that you've included in the study. The cases and controls then provide their exposure histories. And you calculate something called the odds ratio, which is the ratio of exposure odds for the cases that have non-Hodgkin's lymphoma and the exposure odds for the controls who don't have non-Hodgkin's lymphoma. And when this study is done with no sources of systematic error, the odds ratio does estimate the ratio validly. And so the case control design is used very frequently in epidemiology because it's very much faster to start from diseased and non-diseased individuals than it is to recruit 55,000 people and follow them prospectively for 10 or 20 years. So there are a number of study differences between these two types of studies that can affect the validity of results. And I think this is, the most important consideration for people who are looking at the glyphosate epidemiology literature. The first one is recall bias. And so in case control studies, people are reporting on their exposure histories with their disease already established or not established. And the fact of having a cancer can cause people to think a lot about what they've done in their lives that might have caused the cancer. And so there's this concern that cases may report their exposure histories more extensively than controls, which would lead to a spurious association in a case control study between exposure and disease. A second issue is the possibility for selection bias. In case control studies, cases may participate to a higher degree than controls, or controls may be selected in such a way as to not be representative of the case's source population. So there was one case control study, for example, where the controls came from an orthopedic ward, which you wouldn't expect necessarily to be representative of the general population in the area where the non-Hodgkin's lymphoma cases were diagnosed. And in many of the studies, in fact, the proportion of cases who agreed to participate in the study was much higher than the proportion of controls, which raises concerns that you might have selection bias producing a spurious association between exposure and disease. A number of the case control studies had proxy respondents, and as you can imagine, secondhand information is less accurate. And also there's the concern about recall bias may apply to these proxies as well. The proxies were often interviewed because either the case that was included in the study was dead or the case could not, for any other reasons, do the interview for their exposure history. And then the last issue to be considered is any particular pesticide exposure in the farming environment is just one of many exposures. And in general, the failure to take into account in the statistical analysis not only relevant personal risk factors, but relevant other exposures that farmers might have, solvents, diesel exhaust, other things that are more frequent occurrences than a pesticide exposure. Failure to take those into account adequately in the statistical analysis can produce a biased estimate, and we call that to be biased due to confounding. So just to review briefly the validity factors and how they affect the types of studies that were reviewed for glyphosate and non-Hodgkin's lymphoma. Recall bias, since the Ag Health study collected exposure information before the follow-up for health outcomes, there was no possibility there for recall bias. Of course, all 6 case control studies had the possibility of recall bias. Selection bias, there was no possibility for that among the patients recruited in the agricultural health study, but there was marked difference in participation rates in 4 of the 6 case control studies. There were no proxy respondents in the Ag Health study, but there were in 3 of the 6 case control studies. And the confounding control in the statistical analysis was very extensive in the agricultural health cohort study, but by the judgment of our expert panel, poor in 5 out of the 6 case control studies. So here I've summarized the results from the case control studies, and I'll just point out a couple of things about the slides. First, most of the case control studies did their analysis based on even 1 day's use in a lifetime for glyphosate. So you see there are 4 studies where the only analysis had to do with a single day's use or more for glyphosate. But there were 2 studies where they looked at more frequent use than any use. In the McDuffie study, which was a Canadian study, they had an exposure group that they called 2 days a year or more for an unspecified number of years in a person's lifetime. And in the study by Eriksson et al., which is a Swedish study, they had a category of greater than 10 days of use in a lifetime. And you can see both of these analyses of higher use groups in McDuffie and in Eriksson showed statistically significant findings relating glyphosate and non-Hodgkin's lymphoma. And so a lot of the interpretation of these studies have to do with your judgment about whether the validity issues make you question these statistically significant findings. The EPA considered the McDuffie study to be a low or moderate quality study and didn't actually include it in its final evaluation. Iarc considered both of these studies to be worth considering, and in their limited terminology, they can say that these studies have shown positive associations, but chance, bias, or confounding can't be ruled out. And our expert panel didn't consider either of these, actually any of the case control studies, to be high enough quality to be considered. So we didn't think any of the evidence from the case control studies were determinative of a possible association between glyphosate and non-Hodgkin's lymphoma. So let's move on now to the Cohort Study, the agricultural health study. In the 55,000 people followed for an average of about 8 years, there were 92 non-Hodgkin's lymphoma cases. 77% of them reported a history of glyphosate use. And the remainder of the participants in the agricultural health study, 76% of them reported a history of glyphosate use. So you see the relative risk, this rate ratio here is just about 1.0. The agricultural health study also did some more detailed analyses by the extent of glyphosate use that was reported. And here you see they have some frequency of use categories that are much higher than you see in the case control studies, 21 to 56 days and 57 to 1,000 or so days of use. And you can see in both of those instances, the relative risk is actually less than 1, which means the exposure was more likely in persons that didn't have non-Hodgkin's lymphoma than in the persons who had non-Hodgkin's lymphoma. And they reached a very strong conclusion in their article. They said the available data provided evidence of no association between glyphosate exposure and non-Hodgkin's lymphoma incidence. So that brings me to my concluding slide, weight of evidence evaluation for glyphosate and non-Hodgkin's lymphoma. We mentioned earlier on that the epidemiologic studies show that glyphosate is used infrequently. Biomonitoring studies show very low exposure potential. The 3 recent assessments differed only slightly based on how the case control studies were interpreted. Iarc took more of a signal detection approach. They accepted the associations in the 2 case control studies that had higher use categories than any use in a lifetime, with the proviso that the results could be due to chance, bias, or confounding. EPA considered the Eriksson case control study and the agricultural health study to be of high quality. They considered one to show a positive association and one to show no association, and they considered them to cancel out. And the Monsanto expert panel focused on the agricultural health study, cohort findings, as the only study that could be taken at face value, not accepting the case control studies because of all the possibilities for different kinds of bias. So our panel concluded only the agricultural health study is devoid of major bias issues. Those results the evidence of no association reported by DeRuz et al. Are consistent with expectation based on the toxicology and biomonitoring data. So that's it. Thank you.
Thank you very much, John. So we've got a few questions coming up, and I would invite people to contribute those as we're taking them through now. I'll start with one for you, John. So there was an assessment from the North American Pool Project, which was presented at Iarc's 50th anniversary meetings. The authors there said that their analysis suggested that glyphosate use was more associated with an increased non-Hodgkins lymphoma risk. They also state that the larger sample size gives more precise results. Are you in a position to make any comment on those statements?
You know, I've seen the abstract from that study, and you know, when you pool case control studies, what you do is you have a little bit larger sample size, so the random error component of the study is a little bit less, but that doesn't do anything about the issues of systematic error, the recall bias, you know, the selection bias, those kinds of things. And if I take everybody back to Biostatistics 101, you know, when you calculate p-values and confidence intervals, they're calculated under the assumption that you don't have any systematic error. They only address random error. So unless they've done something to deal with the sources of bias in the case control studies, recall bias, which you can't do anything about, selection bias, which you can't do anything about, et cetera, then what they've done is they've just reduced the random error a little bit, more likely to get a statistically significant finding. But again, those calculations are based on the assumption that there isn't any systematic error. So you can't take the systematic error out of the studies, and you haven't really advanced the thinking very much. And of course, the AgHealth study, you know, being a cohort study and not having those sources of error shows a very definitive absence of association. So we would weigh that much higher than a pooled analysis of case control studies.
Okay, thank you. We have a further question relating to the epidemiology, which is whether there were any non-professional applicators that were included in any of the epidemiological studies that were carried out. Again, I don't know if you're able to comment on these.
Right. Well, so in the agricultural health study, they're all licensed applicators. But in the case control studies, they take people in a geographic area. So presumably a portion of the people who report having had glyphosate use are using it in situations other than agricultural applications. So those case control studies probably all had non-professional applicators.
And okay, but I guess probably part of the selection was aiming towards professional applicators, which are more likely to have at least more frequent exposure to glyphosate.
I think if you read the case control studies carefully, they will qualify anybody as exposed if they have a full day's use of glyphosate that they reported in their exposure history. They didn't restrict it to professional applicators. But I would assume that you know, a full day's use of glyphosate would more often be somebody who's using it work-related or otherwise agricultural related, than just somebody who's using it in gardening or something like that.
Okay, thank you. Wally, we've got some questions for you. Are you still on the line?
Yes.
Okay. So Iarc challenged the EU evaluation of the carcinogenicity studies based on EFSA and the German rapporteur's statistical evaluation. Which side of the argument should we believe, and why?
Well, it seems to me that if you take the preponderance of the evidence, it's just overwhelming that Iarc is on one side of the scale and the other regulatory bodies are on the other side of the scale, whether it's the Germans, EFSA, whether it's the Australians, New Zealand, the Canadians, or the Japanese. If you look at it as a balanced argument, the weight seems, in my mind, to be much heavier on the side of the worldwide regulators as opposed to Iarc.
Okay, thank you. And probably one other for you, Wally, here. So glyphosate is pervasive, and human exposure is a broad mixture of glyphosate with many other substances. Wouldn't this increase the risk of carcinogenicity or perhaps other toxicological endpoints?
You know, that's one of those questions that the field of toxicology continues to struggle with, and I think this was pointed out to some degree in John's talk. We're just not exposed to a single agent. If I remember correctly, John, there were 3 pesticides that you guys looked for in the agricultural study. We're all exposed to many different things, and to sort out which one is the actual causative or associated agent is very, very difficult to do. Having said that, the more things we're exposed to, I would think that we have a better chance of having adverse outcomes.
So maybe I'll just say here in the farm family study, of course, even though we timed our measurement around an application, what we actually measured in the applicators would have been glyphosate from all sources, the application and any other sources of glyphosate that they would have, dietary or whatever. So the farm family data that I illustrated on that slide include all sources of exposure, not just the application.
Yeah, plus the fact that they're getting diesel exposure, they're getting other environmental types of exposure, they're getting stuff in their food and in the water that they're drinking potentially. And I think the question was, you know, we're getting complex exposures. Does this increase our risk? And it seems to me logically, yes, it does.
Okay, thank you. One other here now for you, Wally, again. So certain scientific insights suggest that treatment-related tumor formation can already occur at very low doses, kind of doses that are much lower of the ones that are typically used in the kind of OECD protocol studies that you've discussed here. This kind of approach invalidates the hypothesis that a dose response is needed in assessing the relevance of tumors. And it also means that the test rates that have been used in the industry-sponsored studies for regulatory purposes or other research are way too high and completely ignore low-dose effects. What's your opinion on this?
Well, I think that's a true statement, that they do, by design, not look at low-dose exposure. Remember, the OECD guidelines for carcinogenicity studies is to push the exposure because of the low number of animals in the study to see if there's an adverse effect. And these animal studies, remember John's biomonitoring chart that he showed us. The tox data are way up there. The regulatory guidelines are even below that. And yet the human exposures are, what, John, 2-3 orders of magnitude below the regulatory levels? Yeah, that's the chart. I mean, look at here are the regulatory levels, EFSA, the joint WHO, EPA. You see where they are, and the exposure to human, and this is these are applicators, right, and not just the general public.
Well, this includes all biomonitoring to date.
And it's, what, 2-3 orders of magnitude below the regulatory levels, and even further below where the tox studies were done.
at least those levels. I mean, there is a massive margin of exposure between what's been seen in the biomonitoring and the doses used for testing.
Mm-hmm. And the tox studies are designed to find an adverse effect. And even at these very large dose levels, we don't see that with this particular chemical.
Okay. And sticking with the biomonitoring, we've got a further question related to the occurrence of glyphosate in urine. Apparently, there is a recent study in Europe where almost everyone's urine was found to contain glyphosate residues or glyphosate at high levels, whereas the studies that you conducted for biomonitoring, these exposures couldn't be demonstrated. There were a lot of residues that were below the LOQ or were, limit of quantitation, or were at extremely low levels, at least in comparison to consumer exposure. So how representative, John, was the population of the family farm exposure study to the EU population, and why were we likely to see such a disconnect in this way?
Well, my understanding is that the German regulatory authority reviewed all the biomonitoring data, including, dietary or non-applicator exposures, and that the difference between, an applicator exposure that happened, 10 or 15 years ago and the quantification of it, and maybe the same exposure you would get today, given that there's more opportunity for exposure through food or whatever. That would basically be in the same range of the exposures that were seen in the applicator studies, and maybe double them. And what we're talking about are order of magnitude differences that are 2, 3, 4 orders of magnitude difference. So I don't think that additional information takes us into the range even of the regulatory guidelines. Certainly doesn't get us close to that. And of course, the actual amounts of exposure that were included in toxicology studies are a couple of other orders of magnitude higher.
Thank you. Moving back here, we've got a further question for Dr. Hayes. In relation to the conclusions that the compounds do not pose a cancer risk to humans, is this also relevant to the formulated products, which are widely used by professional and non-professional users?
If I remember correctly, the JMPR included both the active ingredient and formulation in their review and said that neither were an issue. But most of what I've looked at has been the active ingredient itself.
Okay, thank you. And I guess that's because what the regulatory authorities around the world request any manufacturer to generate those studies. Okay. So I'm looking now to see whether we've got any further questions that are turning up here. Let's give it one more minute. So if there are no further questions, I would just like to thank our speakers this afternoon for their very comprehensive overviews of both the carcinogenicity and the epidemiological databases relating to glyphosate. There are facilities to ask further questions, which can be found at the Glyphosate Task Force website. All your questions can be dealt with there. And I would just like to draw your attention to our next webinar relating to glyphosate, which will take place on Thursday, the 10th of November, and that will be covering the genotoxic potential of glyphosate. Is glyphosate linked with oxidative stress? And we have two well-recognised experts in this area, Dr. David Kirkland and Dr. Jim Buss. So with that, I'd like to hand back to the webcasting team and thank you for your participation today.
And Caroline, thank you for keeping us on target.
Yes, thank you.